Thank you so much, Ellie, for the introduction. It's my second time at London Calling, and it's always a pleasure to be here. It's such a fun conference. So I'm here to answer everyone's question. Can we do it on FFPE? Just a disclaimer, I cannot do methylation yet, but we can do other things on FFPE. So just to start, I don't have any conflict of interest. So molecular marker testing currently entails detection of copy number variations and single nucleotide variations such as CNVs and SNVs. They currently require large infrastructure costs where equipments can cost hundreds of thousands of dollars, which are high throughput equipment, and each marker requires a different test, so you need to have dedicated lab technicians. Therefore, these are often done, at least in Canada, these are often done at tertiary centers with large genomic facilities. However, they're becoming part of pathology diagnostics, especially in central nervous system tumors such as glioma. Since a lot of smaller and non-centralized labs are becoming interested in pathology diagnosis using molecular marker testing, however, because they don't have the infrastructure, the incomplete diagnosis leads to them referring their samples to tertiary centers and therefore causing delay in patient disease management and treatment, ultimately leading to suboptimal care. The reason why glioma is so well studied in molecular marker diagnosis is because it's the most common malignant primary brain tumor. In fact, glioblastoma, which is the most common subtype of glioma, has a very poor prognosis. It has become a molecular defined entity as of 2016 update by WHO. And there are molecular marker statuses are used in both diagnosis and management of glioblastoma. Our hospital in Toronto, St. Michael's Hospital, has a big neurosurgery unit. So all of our cases are annotated with the molecular alterations, and we all have multiple molecular marker statuses. And this is the complete diagnostic algorithm for glioma in general. This is by College of American Pathologists, or CAP, where each green box corresponds to a different molecular marker requiring a different molecular test. So you can imagine how long it's going to take to go through all these tests. Therefore, there's a need to develop a single test that can simultaneously detect all molecular markers in glioma to reduce turnaround time and complexity. However, as many of us know here, one of the most challenges that we face is that we need a test that works on FFPE tissue because that's the most common specimen type in pathology. And I said we need to develop a test using FFPE tissue. However, as many of us know, with nanopore, whole genome sequencing, or in nanopore in general, we cannot do direct FFPE sequencing. And we have also noticed that with whole genome sequencing, we're achieving insufficient coverage for SNV detection. So to overcome both of these challenges, to make nanopore work on FFPE as well as to achieve sufficient coverage for SNV detection, we decided to use a targeted PCR amplification approach, where we demonstrated feasibility of nanopore sequencing so that it works on FFPE. And we've already published this paper in 2023, so you can read more about the assay. But for today, I'm going to talk about where we are right now, where we have combined copy number variations with point mutations. So everything is pooled in one PCR well plate, and we're sequencing on the nanopore and we have a custom pipeline where we're calling copy number variations and point mutations all on one glioma patient. These are the markers or targets that we're currently detecting with our assay. These are all the markers that are required by WHO for a complete glioma diagnosis and classification. So we're looking at copy number variations for chromosomes, we're looking at gene-level copy number variations, and we have point mutations. I'll be going over briefly about how we have combined everything in one assay, because again, remember, this is a PCR FFPE, so we're not doing direct sequencing, which is relatively easier to do with nanopore to detect different markers. But with PCR, there is a possibility of PCR biases. So for copy number, we've actually adopted copy number losses. We've adopted a SNP microarray approach. I'll be talking about more when I talk about the results. For copy gains, we have adopted to use internal standards. And now let's talk about the results, which is the fun part. So I'll skip quickly over about the point mutation. So the graphs on the right are showing a sample with ATRX and TP53 mutation. This is relatively easy to do, so I'll skip over this. So for gene copy number variations, which I did not talk about earlier, we have, in glioma, there are 3 genes that we need to detect: CDKN2A, 2B, and EGFR. And what we used read depth and log ratios, which is similar to what SNP microarray does. And the sample on the left, or panel A, has a diploid copy of all 3 genes because their log ratios are between -1 and 0.58. And the sample on the right, or panel B, is showing a sample with deletion of CDKN2A, 2B, because the log ratio is minus, and it has a gain or amplification in EGFR. And these are all concordant with our reference results. Now, chromosomal losses. I know this looks a bit complicated, or unless you're familiar with SNP microarray. So what we used is we targeted heterozygous SNPs across the chromosome, and we looked for loss of heterozygosity, indicating a chromosomal losses. So if you do not have any allele frequencies in the gray shaded region in the middle, that indicates there's a loss of heterozygosity. For example, the sample in panel A has a loss of chromosome arm 1P. The sample in panel B has a loss of chromosome arm 19Q. Panel C shows a sample with whole chromosome 10 loss, and panel D, for reference, has a diploid copy of chromosome 10. Again, these are all concordant with the reference results. For chromosomal gains, we actually had to use internal standards because the SNP microarray method was not sensitive enough when we were amplifying FFPE DNA and sequencing on the nanopore. So what we use, we use internal standards with controls in stable areas of glioma. For example, we use 1Q and 19P, which is the opposite of what we expect, and other arms of regions of deletion. And we looked at chromosome arm 7P, 7Q. And for fun, we also threw in EGFR in there. So the sample on the left has a diploid copy of EGFR and chromosome 7, which is indicated by the gray circles. And the sample on the right is showing an amplification in chromosome arm 7P, 7Q, and the EGFR, which is indicated by the red circles. So in total, we have achieved 100% concordance in classification with all of our samples. We have sequenced over almost 100 FFP samples so far. And we have achieved over 90% concordance in marker status. So in blue, I have the point mutation targets. In gray, I have the chromosomal CNV regions. And in purple, I have the gene CNVs. You can see we're still trying to improve our concordance for chromosome 10 and 7 because they are right at this 90% marker. We're trying to get it higher than 90% because why not? And then the last part of my assay, which is trying to find a test or develop a test that reduces turnaround time and cost. So let's talk about the time first. So we have observed it takes us about 3 to 4 business days to go from DNA extraction to data analysis, which from FFP if we're multiplexing a lot of samples. And we have done this on both MinION and P2 Solo. And the cost is less than 300 Canadian dollars, not US dollars. Our dollars are unfortunately cheaper. So when you're multiplexing samples, it's much cheaper to do. And if we're singleplexing, it's still not that high. It's less than $500 per sample. And this includes both the reagent cost as well as labor cost. So now, finally, to wrap up with the significance. So this assay is the first to develop a streamlined approach to simultaneously detect all the different types of glioma markers. To remind you, we're looking at chromosomal gains, chromosomal losses, gene-level amplifications, gene-level losses, and we're looking at point mutations, including the hard-to sequence genes like TERT promoter, which has very high GC content. And we're doing this all on FFPE DNA. By doing so, we have cut down the infrastructure costs and requirements. We've cut down the test running costs and turnaround times. In fact, our current assay time is 1/3 of the combined time cost required by the conventional methods, and our cost is about 10% of all the existing methods. This gives smaller labs the ability to adopt molecular testing, and they don't have to refer their cases to anyone else, which is going to save them weeks or months of referral times. And that's beneficial for both the clinicians as well as the patients. And it's not limited to brain cancer. It also has the potential to be adapted to other cancer types. We have started working on other cancers already, but the data is not ready for a presentation yet. And I'd like to end by thanking my supervisors, my committee members, and everyone, all my funders, and also Nano ONT for having me here, and all of you for your attention. Thank you.